everyone. We have people from KMH and also the other hospitals in Toronto, and more people will join in, but we'll start right now. So today we'll hear from Dr. Stephanie Jones. She really leads the community in relating EMG and EEG signals to the underlying cellular and circuit mechanisms. And she's also this tool that is really useful for the community, the HNN tool, Human Neocortical Neosolver, that can really help people that don't necessarily know so much about modeling to use good models of circuits to try to probe how do they relate to measured signals. And so, yeah, it's my absolute pleasure to invite Stephanie Jones to speak, and you can go ahead.
I'm very glad to have this extraordinary opportunity to describe our research applying biophysically principled modeling of the EEG to guide the interpretation and design of non-invasive brain stimulation. And I'm going to be focusing today on a software we've been developing for this purpose called Human Neocortical Neurosolver. So I'd like to begin by describing the challenge in human neuroscience that we aim to address. We all know that EEG is a very powerful technology because it gives us a direct readout of human information processing with millisecond resolution. But a downside is what we're recording is the so-called macro scale activity that's generated by large ensembles of neurons. And it's still difficult to infer what's going on at the underlying cellular and circuit level. And this circuit level understanding is really critical if we want to use EEG to help us to understand what non-invasive brain stimulation is doing to neural circuits, and ultimately to help us design better stimulation protocols. Now, invasive recordings are ideal for dissecting circuit level activity. There's obvious challenges in doing that. So how do we translate from something we measure outside of the head to what's going on in the microcircuit inside of the head? Well, that's the perfect job for computational neuromodeling, where we simulate the electrical activity of the neurons. And with models, we can have specificity both at this microcircuit generation level and at this macro scale recording level. And this is the primary focus of my research program. We've recently turned our modeling framework into this new software tool that we've designed for clinicians and researchers to begin to develop and test hypotheses on the circuit origin of their human EEG or MEG data. And we're calling it Human Neocortical Neurosolver, or HNN for short. And so in my talk today, I'm going to begin by giving you some of the background on the development of this modeling tool. And I'm going to go through an example of how it can be applied to study one of the most commonly measured EEG signals, a sensory event-related potential. And I'm going to be focusing on a study from my own lab where we've been studying tactile perception and looking at tactile evoked responses. I'm then going to give some examples of how HNN can be applied to study the impact of non-invasive brain stimulation on neural circuits and behavior. And again, I'm going to focus on studies from the lab where we've been attempting to modulate tactile perception using transcranial alternating current stimulation and most recently transcranial magnetic stimulation. So how do we begin to develop a model that can connect these extracranially measured EEG signals to the underlying cellular and circuit level activity? Well, there's two steps along the way. The first is that we have to realize that EEG signals are generated by big electrical currents in the brain. And the first step in getting at the underlying cellular level generation is to perform inverse solution methods that estimate the location, the direction, and the time course of these big electrical currents that we call primary currents that contribute to the recording data. From there, we need to connect these currents to how they're generated at the underlying cellular and network level. And it's believed that these primary currents are generated by the postsynaptic intracellular current flow in these long and spatially aligned cortical dendrites. These dendrites create a big electrical current that creates an electric and magnetic field that's recorded outside of the head and NEG. And again, it's the spatial alignment and synchronous activity in large populations of these pyramidal neurons that creates a signal that would record outside of the head. And so, given that this is where the signal comes from, there are some key canonical features of cortical circuitry that we have to take into account in our modeling in order to study the cellular and circuit origin of the signal. The first is that the cortex is a layered structure. There's excitatory and inhibitory neurons in supragranular and infragranular layers that are synaptically coupled, including these pyramidal neurons in supragranular and infragranular layers that have these long apical dendrites that extend to the layers. These networks don't sit in isolation, but they're constantly receiving input from other parts of the brain. And there are two primary pathways of information flow into the cortex. One pathway, known as feed-forward input, represents the input that brings the sensory information from the periphery through the thalamus and into the cortical layers. The other type of input is what we refer to as feedback input that comes from higher order cortex or non-limbical fluid and contacts directly into the supragranular layers. And then again, the primary current dipole comes from the net intracellular current flow across all of the population of neurons. And now, with this level of detail in a model, the unit of measure of the primary current is current over distance. And this is the same unit of measure that we get out of our source localized EEG or NEG data. And so we have one-to-one comparison between the output of the model and our recorded data. Now, we know something about the size of the networks that contribute to some of the most commonly measured signals, including event-related potentials. These are typically on the order of 10 to 100 nanoampere meters. They're believed to be generated by the synchronous activity of on the order of tens of thousands of synchronously firing pyramidal neurons. Low-frequency oscillations, alpha rhythms, beta rhythms, gamma rhythms, they tend to be larger in amplitude on the order of 100 to 1,000 nanoampere meters, and they're believed to be generated by the subthreshold activity of on the order of 1 million neurons. And so this is the scale of neuromodeling that we built into HNN. And next I want to briefly give you some more of the model detail. So we simulate the pyramidal neurons in this model with multiple compartments. And we have single compartment inhibitory neurons, and they're synaptically coupled to each other through both GABAergic and glutamatergic synapses. We calculate the electrical activity in each compartment of every neuron with Hodgkin-Huxley dynamics, where we have active ionic conductances, sodium, potassium, calcium, in the somatic and dendritic compartments. We calculate this longitudinal current using cable theory. And then again, the primary current dipole signal comes from the net intracellular current flow across all of the population of cortical neurons. And as I've mentioned, it's important to consider that these circuits don't sit in isolation, but they're constantly receiving input from other parts of the brain. We have these two pathways of input, a feed-forward pathway and a feedback pathway. Now, in our model, we don't simulate the activity of all these other brain areas. Instead, we generate trains of action potentials that represent the input from these other brain areas. So for example, to consider feed-forward drive, we could simulate a train of action potentials that activate excitatory synapses in the local network in this feed-forward or projection pattern that we call proximal drive because it effectively hits the proximal dendrites of these pyramidal neurons. This type of input would push current flow up the dendrites to create a positive deflection in our signal. Similarly, we could simulate feedback input as a predefined pattern of action potentials that activate excitatory synapses at distal dendrites. These push current flow down the dendrites to create a negative deflecting signal. And it's a combination of this feed forward and feedback drive in combination with the local circuit activity that creates this signal that represents our primary current diode. Now this is a reduced representation of the network at HNN. The full network contains a scalable number of neurons, and it represents a patch of cortex from a localized brain area. And we've been specifically designing HNN to study some of the most commonly measured signals, including ERPs and low-frequency brain waves. Now, this is a very complicated large-scale network model. All of the code is open source and it's available on GitHub. However, if you don't have experience building and working with neural models, the code can be very difficult to understand. And so in order to make this model accessible to a broad community, we've embedded it in a user-friendly graphical user interface. And so there's no coding necessary to work with this model, where we've created a website that contains installation instructions, background information, tutorials on how to begin to simulate ERPs and low-frequency oscillations, and example data and parameter sets for your work. Now, when you start HNN, this is the graphical user interface that you see. And the top has a schematic representation of the local cortical column model, and it includes these layer-specific pathways from which you can drive the network to represent your simulation experiment. And so to demonstrate how to use HNN in practice, I'm going to go through an example from our lab where we've been using the model to study somatosensory perception and tactile evoked responses. And so in my lab, we perform tactile detection experiments where we give a subject a subtle brief tap to the middle fingertip, and we ask them, did you feel it or didn't you feel it? We record the brain activity with either MEG or EEG, and we isolate the contribution to the signal from the hand area of S1. And so here I'm going to show you in the HNN GUI what the S1 evoked response looks like from a tap to the finger. And in this example, I'm showing you the response from a fairly strong tap to the finger, where the subject reports feeling it 100% of the time. And what you can see is there's a reliable, strong evoked response here with alternating positive and negative deflecting peak amplitudes. And so the first application of our model was to use it to understand how this event-related potential is generated by the underlying neural circuit. So now I'm going to walk you through how we did this. Now, HNN won't automatically tell you how to generate evoked potential. Instead, we think of HNN as a hypothesis development and testing tool. And so the first thing we did is we went to the literature and we said, what do we know about how sensory evoked potentials are generated? And it turns out there's a long history of literature looking at the origin of evoked-related potentials, particularly in somatosensory cortex. And what that literature suggests is that the S1 evoked response is generated by a sequence of activations to the local network that consists of a feed-forward input, followed by a feedback input, followed by a later re-emergent feed-forward. And so in our model, we wanted to test the hypothesis that a sequence of activation similar to what we knew from the literature could in fact generate this macro-scale S1 here. And so what I'm showing you here in the graphical user interface is a histogram of the driving spikes that we use in our network to represent proximal and distal drive. So here we have a histogram of feed-forward drive, followed by feedback drive, followed by this late re-emergent feed-forward drive. We activate the network with these inputs by hitting Run Simulation, and we look at the output of the model, and we see indeed that this sequence of input was able to reproduce the tactile evoked response. And now, importantly, while the literature gave us information on how to begin the simulation. We spent a lot of time adjusting the timing and the strength of these inputs in order to accurately reflect that recorded PRP. And now, with this simulation in hand, we could go in and look at what happens under different experimental conditions. In our initial study, we looked at the evoked response from taps to the finger that were felt and not felt, and tried to infer what are the underlying circuit differences in these different experimental conditions. What I want to describe to you next is how we've used this modeling framework to understand differences in the evoked response that occur from non-invasive brain stimulation. And so to do that, I'm going to now switch to describe some studies in my lab where we've been attempting to modulate tactile perception with non-invasive brain stimulation, state stimulation. And so I'd like to begin with some background motivation for our non-invasive brain stimulation experiments. So in addition to looking at properties of the evoked response, we've been investigating how brain rhythms impact somatosensory perception. And so what I'm showing you here is during the same tactile detection experiment, we're looking at the spontaneous signal from the hand area of S1 before we tap the finger. And what you can see is that there's large amplitude, low frequency oscillation. When we put a frequency filter on this oscillation using a standard wavelet analysis, you see that this oscillation has a strong component in the 7 to 14 hertz alpha band and in the 15 to 29 hertz beta band. And one of the questions that we were interested in is, does the power of this oscillation in the pre-stimulus period before we tap the finger impact the subject's ability to feel that tap to the finger? And it turned out the answer to that question was yes. And so here I'm showing you that frequency versus time representation sorted over trials where the subject felt the tap to the finger and trials where they reported they didn't feel the tap to the finger. You can see very clearly that on trials where they didn't feel the tap to the finger, we had higher power in these alpha and beta bands. And in this way, we think of high power as a signature of inhibited perception. And if we do some statistics and we look at the average, so here we're averaging in the pre-stimulus period across the alpha and the beta frequency bands. We're sorting power from low to high, and we're looking at the detection rate as a percent change in hit rate, as a percent change in hit rate from the mean. So the higher it is, the more likely the subject is to feel the tap to the finger. You can see very clearly there's a linear relationship between power and perception. The higher the power, the less likely the subject is to feel the tap to the finger. And so now, knowing that these spontaneous oscillations are going to inhibit the subject's ability to feel that tap, we next wanted to ask the question, can we use non-invasive brain stimulation in order to enhance these ongoing oscillations, and in doing so, enhance the inhibition of perception? And so to do this, we're going to ask the question, can we modulate rhythms with non-invasive brain stimulation? And if so, can we use H&N to interpret the circuit level effects of the brain stimulation? And we started by looking at these alpha oscillations. There's a lot of examples in the literature that suggest that maybe we can train or enhance alpha activity in the brain and influence behavior. And so the first study that we did, we used alpha frequency transcranial alternating current stimulation. We did our tactile detection experiment before, during, and after 10 minutes of individualized alpha frequency stimulation over the primary somatosensory cortex. The stimulation was on for 6 seconds and then off for 6 seconds. And we compared that to a sham condition where we had just one cycle of a 1 hertz oscillation that ramped up and then turned up. And we looked at the EEG signal above somatosensory cortex before, during, and after this TAC stimulation. It turned out that for various technical reasons, we couldn't look at the EEG during the TAC stimulation. And so what I'm going to show you next is a comparison of this pre-alpha stimulation compared to post-alpha stimulation where we looked at the question, can we enhance alpha activity in our EEG signal? And does that correspond to an inhibition of somatosensory perception? And it turned out with this experimental paradigm, we did not find a significant impact of alpha frequency current stimulation on alpha oscillations in somatosensory cortex. So here I'm showing you alpha power in the sham and alpha stimulation condition pre compared to post. In fact, there's no difference in the alpha oscillation. Next, we looked at perception, and we also didn't find a lasting impact of alpha frequency stimulation on tactile detection performance. And so here again, I'm showing you the percent change in perception from the baseline pre-tax condition in the sham and the alpha condition. And what we found was that during the transcranial alternating current stimulation, we did in fact see a reduction in perception compared to sham. However, this reduction in perception didn't persist into the post-stimulation period. So we didn't have a lasting effect. Now, there were various reasons why our experimental paradigm may not have trained alpha oscillations or impact perception. We were using a very weak electrical current. We had this very brief on and off, and we had a lot of technical challenges with our experiment that are described in our publication on this work. But we did find one subtle but significant difference in our EEG signal, and that was in the tactile evoked response. If we compared the evoked response of somatosensory cortex in the pre versus post alpha stimulation condition, we found that this peak near 70 milliseconds was greater post-stimulation. And in fact, that was a significant difference in the response between 70 and 80 milliseconds. And so this was very interesting to us. And we wanted to see, well, can we use our HNN software to interpret what it is that this alpha frequency stimulation did to the neural circuit to create this difference in the tactile evoked response. And so now I'm going to turn to HNN. Here again is the graphical user interface. I've loaded into this interface the pre-stimulation evoked response here shown in blue. Here is our actual recorded data. This work was done by a graduate student in the group, Danielle Sliva. So I want to thank Danielle for providing this information. And so now we wanted to test a hypothesis about what this alpha frequency stimulation might be doing to the neural circuit. And we started with the hypothesis that maybe what stimulation is doing, while it's not entraining alpha activity, maybe it's impacting the local synaptic efficacy of the connections within the local network. And so we've created an easy way in the software to modulate the gain of synaptic activity between the neurons. And so you can go in to test the hypothesis that this alpha stimulation changes the local synaptic plasticity. And so to do that, you go into this set parameters button. We have a button here that represents the synaptic gains, or the strength between the connections. And you can change them by a multiplicative factor. And so the first thing we did is we said, well, maybe what's happening is that all of the synapses are being strengthened by a factor of 2. And so you can go in and you can multiply all the connection strengths by a factor of 2. This is for the excitatory to excitatory, excitatory to inhibitory, or the inhibitory connections. When we do that, and we hit run simulation and look at the output, we see in fact that an increase in the synaptic efficacy of all of the connections is able to reproduce this difference that we observed in our data. However, it also creates this later difference that we don't observe in our recorded data. And so we have hints that, yes, maybe indeed the alpha stimulation is changing the synaptic plasticity. But next we wanted to see, maybe it's not all of the synapses. Perhaps it's only the excitatory synapses or only the inhibitory synapses. And so we went in next and we checked, well, what if we change only the excitatory synapses and increase the strength of those twofold? And what we found was, in fact, this went in the opposite direction. We got a higher amplitude response, and it didn't match our data. Then we went in and we tested, well, what if it's only the inhibitory synapses? So here we're increasing the inhibitory synapses, by 2 times their strength. And indeed, that inhibitory synapses change was able to reproduce not only this peak difference that we saw near 70 milliseconds, but also the later response to when we saw a lower amplitude throughout this post-stimulus period. And so that suggested to us, the model is suggesting that what this alpha frequency stimulation is doing is it's creating a subtle change in the synaptic efficacy from the inhibitory neurons, and specifically only the inhibitory neurons. Now, excitedly, this is actually supported by some of the literature, which suggests that low-intensity alternating current stimulation preferentially potentiates inhibitory synapses. And so now we have some evidence from our model that that may be what's underlying the difference in the EEG that we observed. Now, I've been showing you the macro-scale current dipole signal. But one of the powerful things in modeling is that we can go in and we can look at all of the microcircuit activity. And so we've created a way in HNN to go in and to view the microcircuit details. So if you go under view, you can look at layer-specific dipoles, you can look at spiking, you can look at frequency responses, somatic voltages, and different things in the network. So here I'm going to show you the spiking activity in the network. And so here's the spiking activity as a raster plot in all of the different cell types in our network before we change the synaptic plasticity, representing our pre-tax condition. And once we increase the inhibition twofold. And what you can see is that we increase the strength of inhibition, we get less spiking in our pyramidal neuron populations. And this decrease in spiking corresponds to a lower amplitude evoked response. And so now we have some very specific predictions about what's happening in the underlying circuit. And we can then test those predictions with key-based reporting or other imaging modalities. And we have some targeted directions to go in and further test what this brain stimulation is doing to the underlying neural circuit. And so this was very exciting for us, but in our hands, what we wanted to do was modulate tactile perception. And this experimental condition did not modulate tactile perception in our hands. And so the next thing we did is we went back to our data. And when we looked more closely at our data, we found that, in fact, the beta component of this oscillation is more prominent in somatosensory cortex. And in our hands, it's a stronger predictor of tactile perception. So next, we set out to see if we could instead modulate the beta component of this rhythm, and in doing so, again, inhibit tactile perception. But before doing that, we took a step back to understand our data a little bit better. What I'm showing you here is average data. We're averaging across time, we're averaging across frequency span, we're averaging across subjects. But the brain isn't averaging. And so next we looked more closely at the unaveraged response. And so what I'm showing you here is 10 examples of the pre-stimulus activity before we give that tap to the finger. And this is the corresponding frequency spectrum. And what you can see as these beta oscillations emerge as these transient events. They typically last less than 150 milliseconds. Now, because in the frequency domain, these signals are always positive, when you average in the frequency domain, you get an accumulation without cancellation. And you get this continuous band of high power activity. And you might automatically think, oh yes, there's this continuous ongoing oscillation that's happening before we tap the finger. When in fact, at any moment in time, you either do or do not have one of these transient beta events. This transient nature of beta activity is a very robust phenomenon. We've been fortunate to be able to look at it also in local field potential recordings from the rodent somatosensory barrel cortex. This is data provided by my collaborator at Brown University, Professor Christopher Moore, and a graduate student, Heian Shin. And in the mice, they're doing very similar tactile detection experiments where they're stimulating the vibrosa. And here we're looking at the spontaneous activity in the somatosensory cortex, again, before the tap to the finger or the tap to the vibrosa. You can see that there's very similar expression of activity. We've quantified various features of these beta events in the pre-stimulus period. We sorted the event number, we looked at the event power, the event duration, frequency span, and we found remarkable homology across not only species, but these different reporting modalities that suggests that this transient nature of data is a very robust and reliable phenomenon. This has now also been observed in motor cortex, striatum. Transient beta activity seems to be very consistent across many different brain areas in paradigms. I point briefly to some code that we have on GitHub where you can begin to start to analyze your signals and measure various features of transient oscillations. So now that we know that these rhythms are continuous, we have to revisit the question, what features drive this relationship between pre-stimulus power and detection? Remember, if we had low average power, then the subject was more likely to feel that tap to the finger. If we had higher power, the subject was less likely to feel that tap to the finger. And so when we think of an oscillation being higher in power, we might automatically think, well, that means the oscillation was higher in amplitude, or there was a bigger network contributing to the response. But now that we know that these oscillations are transient, there's several features that can contribute to a difference in average power. It could be that in one case, so here I'm just showing a schematic representation, frequency over time. In one case, in the pre-stimulus period, you have one event versus two events that are at the same amplitude. When we average this, we'd get high power, and over here we'd get low power. Or it could be that in both cases we have one event, but in one case the power of that event is higher, and so when you average, you get higher power in the average signal. Or it could be that the duration is longer, or that the frequency span is extended. Any one of these differences could underlie this difference in power and its impact on perception that we've been seeing. And it turns out that it's actually the number of events in the pre-stimulus period that contributes to higher power and inhibited detection. We also found that the timing of the event before you tap the finger mattered, such that the closer it was to the time of the tap to the finger, the less likely the subject was to feel that tap. And the most influential period was approximately 300 milliseconds before the tap to the finger. And this work has been published. I don't have time to get into further details. We've also used our HNN modeling framework to try to understand the circuit origin of these transient beta events. And the modeling, combined with invasive recordings in both rodents and non-human primates, suggests that these beta events are coming from a strong burst of excitatory input to supragranular layers. And again, I don't have time to go into details, but for further details, you can look at our publications on this work. Okay, so now that we know these rhythms are transient, we have to re-ask the question, how might we apply non-invasive brain stimulation to more accurately account for these transient rhythms? And so what we're doing now is we're trying to adapt a new way of thinking, where we're developing brain stimulation paradigms aimed at mimicking the endogenous expression of these transient rhythms. And in my lab, we're currently switching to TMS, where we have more control over the pattern and the timing of the inputs. And we're developing a protocol and asking the question, can TMS bursts of TMS pulses to somatosensory cortex that mimic this endogenous pattern of beta activity, can they be effective at inhibiting perception? But of course, there's a major issue that we're having to deal with, and that TMS is not like endogenous brain activity, because the device itself drives sensory responses. You can feel the stimulation, you can hear the stimulation, it's driving muscle activation, and we now know through many studies that this can be reflected in the TMS evoked EEG activity. So there are lots of ways to minimize these compounds, but we don't yet have a way to remove them. So instead, what we're doing is we're trying to see if we can use HNN to help us understand this compound. And specifically, we're going to ask the question if HNN can help us understand if and how the sensory component of the TEP, TMS evoked potential, is reflected in the EEG signal. And so now I'm going to briefly show you some preliminary data from the lab, where we've been recording EEG from bursts of TMS pulses to somatosensory cortex. This work is being led by the graduate student in our group, Danielle Sliva, and also a postdoc, Carmen Cole. They're going to be presenting a poster with more details on this work in a session this afternoon, so please tune into that poster for further details. So in this experiment, we're using MRI-guided TMS to give bursts of TMS pulses to somatosensory cortex, and we're recording the response in an electrode above S1. And so here I'm showing you in our HNN graphical user interface the response to that burst of 3 pulses to somatosensory cortex. And these pulses are separated by 20 milliseconds. And you can see that this creates a very strong and reliable response in the somatosensory cortex. Now here we've masked the noise from the TMS stimulator with earplugs, but the subject is most definitely feeling the sensation of this TMS burst of input. And an important thing to note is that this TEP is remarkably similar to the tactile evoked response in S1 from that tap to the finger that I showed you earlier. So it suggests that at least part of what we're seeing may be reflecting the sensory information from this input from our TMS device. And so recall also that we were able to use HNN to look at the circuit origin of this evoked response by driving the network with a sequence of input. And so next we wanted to ask, can a similar sequence of input reproduce what TMS, including the sensory component of TMS, is doing to reproduce this EEG response that we recorded? And so what we're going to do to test that hypothesis is we're going to simulate a sequence of drive similar to the sequence of tactile input, but here we're going to do it 3 times because we're giving 3 pulses of TMS. And so we have this feed-forward, feedback, feed-forward input that's very similar to the input that creates the sensory component of a tactile input to the fingertip. And we're going to hit one simulation. And in fact, we see that remarkably, the sensory evoked input can almost perfectly reproduce this TEP that we're seeing from our TMS device. And so that suggests that this evoked response is at least in part reflecting the sensory information. Now, importantly, this earlier component of the evoked response may not be influenced by the sensory information. And so what we're doing ongoing in the lab is looking more closely at the earlier components of this evoked response. And again, for further details on that, please tune in to our poster later this afternoon. And so with that, I'd like to conclude. I've described our new software, HNN, which represents a neocortical column model under thalamocortical and cortical drive that represents this feed-forward and feedback drive to the input. It simulates the primary electrical currents that underlie EEG and MEG with enough detail to connect the macro-scale recording to the underlying cellular and circuit level activity. HNN is now developed into this graphical user interface. It's a user-friendly software which you can download and get all sorts of information from on our website. And I hope I've convinced you that HNN can be applied to understand the impact of non-invasive brain stimulation on neural circuits through the corresponding changes in EEG signals and can aid in the development of new brain stimulation protocols. So with that, I'd like to thank my collaborators. I'm fortunate to have an amazing lab at Brown, and everyone has contributed to this work in a meaningful way. The HNN software is being developed by a large network of researchers at different institutions, including Brown, the Martino Center at Mass General, and Yale University. And I'd also like to thank my collaborators at Brown and the Nathan Klein Institute in Columbia, who have been kind enough to share their animal recorded data to begin to test some of the predictions that come out of our modeling software. And so with that, I'll leave you with the conclusions and a reminder to please check out our poster later today. And I thank you again for your time, and I'll be happy to answer any questions in the Zoom session that follows. Thank you.
So just a reminder, you can post your questions and Stephanie will answer them. I can start with a question of my own. So it's very interesting to see the TMS use for teasing apart the roles of different parts of the circuit. I do obviously really like the work you did with actual stimuli. So maybe you can say a little about where do you think the TMS will really add a lot compared to, you know, because this tactile stimuli are pretty high resolution as well in terms of time, right?
So, well, first of all, thank you, Itai, for inviting me to speak, and thanks everyone for allowing me to use this talk that I already created rather than repeat myself for another hour. And I hope that it came through clear enough.
Yeah, so I'll just say one thing. Stephanie is an amazing speaker, and there is a recording of this talk on YouTube, which we will send to you guys and girls, and you can see it well after if there were any parts that were a little choppy. So thank you, Stephanie, for joining us. And yeah, the link is right here in the chat.
Okay, excellent. And I'm happy to answer questions at any time if people want to email me. But can you repeat, Ita, your question about the TMS?
Yeah, so you've been probing the circuits both in response to stimuli, actual tactile stimuli, and also in response to TMS. And each method can offer, obviously, it has its pros and cons, right? So maybe you can tell us a bit more about where do you see the TMS adding to the story?
Yeah, it's been an interesting path because, you know, our goal with the TMS was to see if we could probe the circuits and change somatosensory perception, ultimately hoping to move towards pain perception. But then we started integrating TMS and EEG into the lab and realized there's lots of issues with trying to do TMS and EEG. And one of the main issues is this sensory artifact. And so when we tap the finger, you see this very reliable SEP. When you tap the somatosensory cortex with TMS, you see almost the same exact signal. And so then the question becomes, is that really because the subject is feeling the tap on their head, like a tap to the finger, and so it's the sensory component of stimulation? Or is it really because once you drive the somatosensory circuit, you're engaging the same circuit that you're engaging when you tap the finger, right? You're driving the somatosensory cortex, it's going to the thalamus, it's coming back up. You're doing the same thing if you drive spikes directly from TMS that you would from the thing. And so now we're trying to tease that apart in a better way with more realistic sham coils, doing some clever tricks with our experiments to try to remove that sensory component. The other thing is, we want to mimic these beta events, which we're realizing we don't want to drive oscillations because that's not what we're seeing in our data. But when we're trying to say, oh, we can reproduce this large scale sub-threshold beta oscillation, it's really hard to know what's the right strength of TMS to use for that, right? Because TMS is driving lots of spikes. And we don't think these beta oscillations are driving lots of spikes. We think it's a largely sub-threshold activity. There's, of course, some spiking underneath. But the balance of how to recreate these things that are happening naturally with this device, there's a lot of challenges there. But I really think it's a powerful way for the field to be moving forward, particularly for my group that's doing macro scale EEG cortical stuff. It's a natural growth direction to try to understand what brain stimulation is doing to these circuits through the lens of what it's doing to the EEG.
Interesting.
We're excited, and it is new for us. We've been doing this for the past year and a half or so.
Yeah, we have a lot of people that are interested in seeing what it leads. Thank you. So a question from Dr. Francis Skinner. Thank you for the presentation, Stephanie, and nice to see you again. Oh, she's just saying go around now. Actually, it's not a question.
Thanks for coming, Francis.
Okay.
I'm sorry,
Jessica,
not in person.
Yeah, we have a nice attendance from that group. Alex from our center is asking, are contributions from deep layers to EEG meaningful in comparison to contribution from more superficial layers? If so, do different layers contribute to different frequency components of the PSD in the EEG?
Yeah, it's a really interesting question. And I think a lot of it depends on the experimental paradigm and the state in which you're recording. When we're using HNN, what we're actually simulating is the primary current dipole. And so when you apply inverse solution techniques to your EEG data, you're estimating what's the big electrical current that that's generating the signal where you record outside of the head. That's going to have contributions from both layer 2, 3, and from both the supragranular and from the deeper infragranular layers. Because the pyramidal neurons in the deeper layers are longer, they contribute more to the signal. And so with the modeling framework, you can start to play games as, okay, let's suppose I only have layer 2, 3 active. How big would the signal be compared to if I also have layer 5 active? And how much of the network, you could have a smaller layer 2 dipole, but activated over a larger area. And that could compensate for a layer 5 dipole, but over a smaller area. And so you have to think about how all these things relate. And ultimately, you can use the modeling to make different predictions. And then you want to go test those predictions invasively or with other imaging techniques. There's now this laminar MEG analysis that's being created at UCL and in some other exciting GABA spectroscopy and different things we can do in humans. But ultimately, modeling is one tool to try to tease apart the contribution from the supragranular and infragranular, where the different frequencies are coming from. And I don't think there's one answer, that high frequency oscillations are coming from supragranular and lower frequency from deeper. I think it's going to depend on what your experimental paradigm is, where you're recording, what's the brain state at the time.
Great. Frank, you can probably read your question.
Yeah, sure. Yeah, great talk, Stephanie. I was just wondering, maybe I missed it, but have you found any intraband differences in the transients or power of the beta events, specifically like low versus high beta?
So have we found, interband differences? So it's interesting, we study adults and you really tend to get one high power band. And it's usually between, and again, we're in somatosensory cortex, and it's usually between 18 and 23 hertz. And if a subject is in that band, they kind of stay in that band. And so we don't really see there's a low power and a high power band. It's really just one main high power regime that we're looking at. So no, we haven't really found anything in interband interactions. We see a lot of interaction between the alpha band and the beta band, because in the sensory motor cortex, there's a clear distinction between what's less than 15 hertz and what's greater than 15 hertz in this 20 hertz band. And so we have seen some differences there, but not within, say, 15 to 29 hertz. We don't usually see that in our data.
Okay, very interesting. Thank you.
I'll give some time for people to ask. In the meantime, about your transient events, which we find also very interesting because it's not really one continuous oscillation, right? Do you find that they are similar, like those events? They just, of the same kind of look and type, they just occur in different times, or is there a change in some feature of those events?
Yeah, we've been looking really hard at that to see, like, are there features of the waveform that differ with attention or that are predictive of perception? And what we're finding is, at least for the really high power ones, and so we do this thing where we threshold and we're only looking at the ones that are really contributing to the power that we see in our signal, they're remarkably stereotypical. They tend to look the same. They look the same in somatosensory cortex, motor cortex, frontal cortex. It suggests that there's really something kind of macro scale-ish about the structure of the cortex that's dictating where these oscillations come from. We have started looking at changes in the beta events with age, and we have seen a progression with age of the shape of the waveform. And so that may be something interesting that's happening with age. We have one pilot study where we show that the beta power increases with age, and we just started to look at how is that related to the waveform shape or the number of events. And there is a recent paper showing that it is the number of events that changes with age, but there may also be some changes in that waveform that we're seeing.
Great. A question from John Griffiths. Do you think excitatory versus inhibitory plasticity induction from TACs and excitatory versus inhibitory plasticity from RTMS, including theta burst, can be understood as reflections of the same phenomenon, or are they quite different?
Hey, John. Thanks for coming to the talk, and thank you for that question. It's really interesting. I don't know. I think, there's lots of inhibitory neurons towards the surface of the brain. And if what TACs and TMS are doing are driving those inhibitory neurons in layer 1, then certainly they could be doing similar things. I think one way we might be able to answer the question is, by doing the same experiment, like this finger tap that we're doing, before and after TACs versus before and after rhythmic TMS, and seeing, does it influence the tap the same way, or whatever experimental paradigm you're doing? And can we mimic that in our modeling with changing the excitatory versus inhibitory plasticity in the same way? But TACs and TMS, they're slightly different beasts, because TMS is driving spikes, where TACs is more of this sub-threshold neuromodulatory effect. And so there's a world in which they could be doing different things. But again, if they're really hitting the same circuits ultimately, like the inhibitory neurons, then they might be more understood as reflecting the same thing. So short answer is, I don't know.
Frank has another question, and Frank, you're welcome to read it.
Yeah. So I was just wondering about the transient beta events, and specifically the power and how the power changes. So would you say they consistently have a sudden increase in power that's sustained and then a sudden decrease, or is it a gradual increase and decrease? And is it sort of similar between events, or do you see differences in the sort of power profile of the higher power events?
So mostly what we're seeing in our data is the higher power profile. So if we see higher power of a single event, right, that's usually because if we were to compare event to event to event, and one event had higher power than the other event, it's either because it's more sinusoidal, or because we're putting sinusoids to calculate power, or it's because you have a higher amplitude oscillation. And so those are two different ways that a single event, when you compare event to event, could have higher power. But mostly when we're looking at high power, we're averaging. We're averaging across time, and we're averaging across trials. And in that case, we find more often than not that the difference is coming from having more of these transient events than it is having more transient events that are higher in amplitude or more sinusoidal. But again, I think it may depend on the experimental condition, right? We're looking in somatosensory cortex, we're looking at shifts in attention pre-stimulus periods. It may be different if you're looking at beta oscillations that are driven by sensory stimuli or that are rebounding after desynchronizing. We haven't looked at any of that yet. And so I think any of those features could be relevant and important. And I know in Parkinson's disease, there's now some evidence that when you get higher power in Parkinson's, it's not only that you get more of these events, but they do get sharper in amplitude. And so that contributes also to the high powerness, is that the events have a sharper waveform. And then when they give DBS, that sharpness goes away. And with it, the high power is also diminished.
Very interesting. Yeah, thank you.
I think you're muted.
Yeah, I'm muted. Thank you. What's interesting is that we've been looking at aging effects on EEG from a different angle, because we have some electrophysiology finding for differences in cellular features that we then propagated through the circuit. And your comment about that when there is more power in beta, there is less detection is very interesting, because we see something somewhat similar, an increase in beta in those age circuits, or in the ERP at least. And maybe, is it because maybe the circuit is more noisy? What's your interpretation of that?
So we did a little bit of modeling on this. We have a 2010 paper, Ziegler et al., Neuroimage, where we looked at where this and again, it was like a pilot study of, I think, 12 subjects where they were between 18 and 45. So it wasn't elderly, it was middle aging. And what we showed is that we could get the increase in beta by changing the synchrony of the inputs. So if the inputs, the way we get these beta oscillations is we have our local cortical circuit and we drive it with inputs. And if we made those inputs more synchronous, then we could reproduce features of not only the power, but the influence on the ERP that we saw, which also changed with aging. Now that we're quantifying in more detail the event shapes, it really seems to be more about that distal drive. And if we increase the synchrony and the strength of that distal drive, that's what can create the downward deflection. The other thing that is interesting is that driving interneurons specifically can have unique influence on that dipole current. If you drive interneurons on the supragranular layers, that's going to pull the current up. If you drive interneurons in the deep layers by the soma, that's going to pull the current down. And so if we have more drive to that somatic targeting inhibitory neurons, that could also create that downward current flow. And so now there's two ways that we could get a stronger peak. One is drive excitation greater, which is going to push current flow down. But the other is drive inhibition at the soma greater, and that will also pull current down. And so some kind of a balance between those that we're thinking about now.
Interesting.
And that really targeted, invasive recordings would help distinguish.
Excellent. So I think we are done with questions. And it's been a real pleasure. The people will have a link to your presentation as well to hear it again.
Excellent.
And yeah, thank you so much for joining us. And it's nice, at least, even though the era is a bit strange these days, that we can connect remotely online and still communicate, right?
Yeah, it's really been one of the pluses of this whole pandemic is the amount of community that we can build, because we're all, happy with sitting in our home offices and connecting and not having to travel across.
Yeah, save some of the logistics.
Exactly. And like I said, thank you again for the opportunity. I'm happy to answer questions that people have if they get a chance to watch the talk again later and some questions come up.
Excellent.
And if anybody's interested in using HNN, please be in touch with us. We're eager to help people get started. And improving our installation and tutorials all the time, and the feedback from the community really helps there. So
yes, we'll send this talk link to people around CAMH. I think it will be really useful for a variety of users, and it looks like a very attractive tool. So thank you again.
Excellent. I hope so.
All right. Take care.
Stay well, everyone.
Yes. Bye-bye.
Bye.